Hello students, welcome to the EPG Pathshala. I am Dr. M.N. Gupta, Professor Emeritus from Department of Biochemical Engineering and Biotechnology at Indian Institute of Technology, Delhi. Today we are going to talk on module Hypersensitivity 1 from paper Immunology. Now we move over to discussing responses of the immune system, which mostly occur during the second encounter with the antigen. So the immune system is primed, so to say. In many cases, the overreaction results in tissue damage. Hypersensitivity reactions are of various types. Today we will look at their classifications and discuss one type of hypersensitivity reactions. So the objectives of this module are to briefly acquaint ourselves with what is hypersensitivity and why it happens. Then to know about classification of various types of hypersensitivities, and to understand allergy in more detail today, which is type 1 hypersensitivity or IgE mediated hypersensitivity. So hypersensitivity reactions we will see in terms of the concept map, that it can be of 5 types, and today we will focus on the type 1, which is allergy, IgA mediated type 1 hypersensitivity, and we will look at the causes of allergy, its clinical manifestations and mechanism. Under certain conditions, immune system overreacts, and this happens because it's making effort to protect the animal. This results in damaging tissues. In certain cases, this may even be fatal. In early years, hypersensitivity was used as a synonym of allergy. So, investigating allergic reactions led us to realize that hypersensitivity is not just about allergy, it can be of various types. So we now call allergy as a type 1 hypersensitivity. The earlier books and the literature lists anaphylactic reactions only under type 1 hypersensitivity. In a fast developing area like immunology, reading the earlier literature sometimes can cause confusion. Today, anaphylactic reactions now covers all allergic reactions. Hypersensitivity reactions are antigen specific. Apart from inappropriate responses to microbial antigens, hypersensitive reactions also occur against self antigens. So autoimmunity in a way is a subset of hypersensitive reactions. Hypersensitivity is not the first response to an antigen. It occurs if a previous exposure has taken place. Hence, in many cases, these adverse reactions originates from or involve memory lymphocytes. Nevertheless, it should also be pointed out that hypersensitivity is a part of normal immune response. It is in a way clinical manifestation when immune response is localized and or the large amount of antigen is involved, because when the large amount of antigen is involved, that leads to also involvement of the large amount of antibodies or immune cells. Let us start with the classification of various types of hypersensitivities. This table shows the classification, the time period in which clinical manifestations occur, and which components are involved. These have formed the basis of classification of hypersensitivities. Gale and Coombs divided hypersensitivity into 4 classes. Later on, a fifth one has also been created. It is necessary to realize that such a classification helps in efforts to understand these reactions. In reality, in any case, more than one type of hypersensitivity can be involved. As we know from our common understanding of allergy, not all individuals react the same way to an antigen, which in this case is called allergen. Environments, genetics, and age all play important roles. Some cases involve external antigenic substances, which may be harmless in the case of others. Type 1, 2, 3, and 5 all involve interaction of antigen and antibody, and the response is fast. That is, speaking relatively to type 4 hypersensitivity response. Type 4 hypersensitivity depends upon T cell mediation and takes much longer time to manifest. Hence, this is often referred to as delayed type hypersensitivity. Gale and Coombe did recognize some distinct features of type 5 and had referred to as stimulatory. This is by some people treated as a separate class and called type 5 hypersensitivity. Type 1, which is also called immediate hypersensitivity, this response is seen within minutes and hence is also called, and that is the reason it is called immediate type hypersensitivity. Here IgE isotype antibodies are involved. Mast cells release vasoactive amines and chemokines which mediate acute inflammation. Type 2 is also sometimes called antibody mediated, as antibody dependent cytotoxicity by NK cells or macrophages or complement mediated lysis is involved. Type 3 is also called immune complex mediated type. Type 5 involves antibody binding to a cell receptor such as a hormone receptor to switch on the cell. Type 1 hypersensitivity, commonly called allergy, as we mentioned, is mediated by IgE. It is believed that IgE evolved to protect us from worms. The immune mechanisms which operate against bacteria, viruses, fungi, protozoa, and worms differ. Bacteria and fungi which do not invade cells are opsonized by the specific antibody. Phagocytosis or complement-mediated lysis takes place. Microbe with intracellular habitat, viruses, some bacteria and protozoa are taken care of by TC or NK cells. Fungi require initially Th1 response. If the fungal infection is severe or persists, Th2 cells respond. Both B cells and T cells are involved in fighting against protozoa. That is why vaccine development against protozoa caused diseases is difficult. Worms, which are also called helminths for example, vary in their sizes and complexity. Infection by worms is responded by class switch to IgE and eosinophil cells, mast cells, macrophage and CD4 positive T cells. It is the Th2 response which results in class switch to IgE. While the size of worms makes it difficult to deal with them via phagocytosis, IgA, IgE and IgG can opsonize. This allows macrophages, eosinophils and mast cells to bind via Fc receptors. Eosinophils and mast cells degranulate and release their contents. The incidence level of allergy in the industrial world of the developed countries is as high as 30% of the population. The percentage is less in developing countries like India, but unfortunately we are catching up fast. This trend is not easy to explain, so we should not hurry and oversimplify issues. In UK, percentage of the some diseases like asthma, allergic rhinitis and atopic dermatitis seems to be reaching a plateau. Others like urticaria, food allergy and angioderma seem to be on the rise. Ironically, the poor hygiene may correlate with lower incidences of allergy, and this is known as a hygiene hypothesis. This may be due to increase in Th1 response in these individuals. We will talk about the sensitization phase little later in this module to understand this. We have used the word atopic dermatitis while discussing the trend of allergic incidences. Atopy actually is used for conditions which display type 1 hypersensitivity such as asthma, hay fever and eczema. Increased risk of atopy is associated with gene loci such as HLA, the cytokines IL-4, IL-5, IL-10 and IL-13 specifically, and leukotrienes LTR1 and LTR2 receptors and chemokines CCR3. We will discuss it all in more details later on. In Western countries, 40% of the population are prone to show IgE response to common environmental allergens. Atopy can also be called this situation and is marked by higher levels of IgE in the circulation and higher levels of eosinophils than non-atopic or the normal individuals. This slide shows the IgE production during allergic responses. IgE is produced by plasma cells in lymph nodes which drain the site of antigen entry or at the local sites where allergic reactions occur. In the latter case, the germinal centers within the inflamed tissue has plasma cells which release IgE. Unlike other isotypes, IgE is mostly found in tissues in bound form to mast cells, which have high affinity receptors for IgE. These are called Fc epsilon R1. Basophils and activated eosinophils also have Fc epsilon R1 and hence also take part in type 1 hypersensitivity reactions. This table lists the variety of allergens which commonly causes allergic reactions. Broadly classified, allergens can be pollens, they can be insect venoms, they can be of microbial origin. Allergens can also arise from animals like hair or denders. They can also arise from the food material like nuts and seafood. In some cases, the drugs, notably penicillin, salicylates, and anesthetics, are known to cause allergy. Allergic rhinitis, commonly known as hay fever, is the most common allergen in United States of America, and there it affects 20% of the population. The causes are grass and tree pollens, systemic release of worms, Echinococcus antigen from hydrated cysts can cause anaphylaxis, which we will discuss shortly. About 300 million people are affected by asthma globally. About 2.5 times more incidences occur for children as compared to adults. Allergic asthma can be caused by a variety of allergens. At early stages, it is mediated by IgE. This slide shows the nature of the allergens. Many allergens enter through inhalation. Many of these are proteins. Inhalation of all proteins, however, does not cause allergy. Enzymatically active proteins, notably proteases, are often allergens. Allergen proteins tend to be highly soluble proteins which do not have very high molecular weight. Pollen grains and mite feces act as their carriers. In the mucosa of the airway, these dissociate from carrier and diffuse into mucosa. The feature of the inhaled allergens that promote the priming of Th2 cells that derive IgE responses also involve their stability. Allergens can survive in desiccated particles. In the low doses, they favor activation of interleukin 4 producing CD4 T cells. Because of the low molecular weight, allergens can diffuse out of particle into mucus. They are also likely to contain peptides that bind host MHC class II. That interaction is required for T cell priming. Let us look at the IgE class switching in B cells, which is initiated by Th2 cells. This slide shows in the first panel, in the first step, activation of NK 1.1 positive subset of CD4 T cells. Native T cells are primed and develop into Th2 cells in the presence of IL-4. Finally, activated Th2 cells secrete interleukin 4, inducing isotype switching to IgE. The main APCs in the respiratory mucosa are myeloid dendritic cells. After processing antigen, these migrate to local lymph nodes, become professional antigen processing cells with co-stimulatory activity that favors Th2 cells. Only very low dose of antigen is involved. For example, in whole of an year, total exposure per person to ragweed, which is the common name for Artemisia artemisifolia, is less than 1 microgram. These small doses are enough to cause Th2, causing class switching to IgE isotype. In fact, allergens can be defined as antigen which provoke Th2 cells to cause class switching to IgE. IgE production requires Th2 and is inhibited by IF and gamma producing Th1 cells. We have learnt earlier that low doses of antigen favors Th2 activation rather than Th1 activation. IgE is important in host defense against parasites. IgE mediates activation of accessory cells which play an important role in resistance to parasites. Mast cells are located near the body surface and are able to recruit both specific and non-specific components of the immune system near the immediate site of entry of infectious agents. They can cause muscular contraction which can lead to expulsion of pathogens from lungs or gut. In helminth infection, mast cells accumulate in intestine. This is known as mastocytosis. Infection by parasites, especially helminths, leads to increase in eosinophil count in blood and tissues. This is known as eosinophilia. Eosinophils obviously have a critical role in defense against helminths. If polyclonal anti-eosinophil antibody is injected to deplete eosinophils, it increases the severity of the infection by worm Schistosoma mansoni. Microscopic examination of infected tissue show degranulated eosinophil bound to the worm. The binding, as explained before, is mediated by opsonization by specific IgE, IgG or IgA antibody. Similarly, blood-sucking ticks provoke participation of IgE, mast cells, basophils and eosinophils. The skin near the tick bite shows degranulated mast cells and accumulation of basophils and eosinophils that are degranulated. Many parasites during invasion of the host secrete proteolytic enzymes for breaking down connective tissue for gaining access These enzymes promote Th2 responses. Many enzymes, in fact, are allergens. Dust mite is responsible for allergy in 20% of North American populations. The allergen is present in its feces and it's a cysteine protease called DER-P1. DER-P1 is homologous to papain. It's a non-specific protease and commonly used as meat tenderizer. The people isolating it routinely at a large scale develop allergy. Such allergies are called occupational allergy. One major application of industrial enzymes is in detergents. Cetirizine, a bacterial enzyme, is routinely incorporated in detergents. Its inhalation is known to cause asthma. The first generation of enzyme-based detergents decades back were withdrawn from the market as large incidents of allergy in housewives were reported. Coming back to DER-P1, it cleaves occludine, which is part of intracellular tight junctions. The DER-P1 breaks the integrity of epithelial tissue and reaches subepithelial antigen processing cells, mast cells, and eosinophils. Papaya latex also contains another protease called chymopapine. It is used to treat patients with sciatica by hydrolyzing protein component of intervertebral discs. A major complication, although fortunately rare, is the acute systemic response called anaphylaxis. Enzymes are being increasingly employed in place of chemical catalysis, or chemical catalysts rather. In general, these are considered greener options. However, this must not be confused with the safety. All enzymes are proteins and proteins are good antigens. Hence, safe handling of enzymes is required in all occupations wherein they are routinely handled and often in larger amounts. One should especially avoid their inhalation as enzyme powders, which are actually the commonest form of enzyme preparations. Enzymes are not the only allergens. Many plant proteins are known to be good allergens. Two allergens from filarial worms are enzyme inhibitors. While it is true that inactivated papain when injected does not provoke IgE response, no systematic association between enzyme activity, biological activity of the allergen, and its allergenicity has been found. Typical allergens are about 3 to 30 micrometer in diameter. Pollens, house dust mite feces, fungal spores, animal skin flakes are common allergens. Included in small quantities, inhaled in small quantities, these adhere to mucous membrane of nasal passages and airways. This table shows the route of the entry of some allergens and its consequences. The symptoms thus depend upon whether the allergen is inhaled, swallowed, or injected. For example, the syndrome systemic anaphylaxis. In this case, the common allergens are drugs, serums, venoms, and even peanuts. The route of entry is intravenous. Response normally is edema, increased vascular permeability, tracheal occlusion, and circulatory collapse, and even death. Similarly, for acute urticaria, which is in common parlance called wheal and flare, the common allergens are insect bites. Route of entry is subcutaneous, and it results in the local increase in blood flow and vascular permeability. Food allergy, in fact, is more common than one would imagine. Tree nuts, particularly peanuts, shellfish, even milk, eggs and fish are known to act as allergens. Their route of entry of course is oral. The clinical manifestation in such cases is vomiting, diarrhea, itching, urticaria, and in some cases, very rarely, it can even known to cause anaphylaxis. The allergic reactions can be discussed in terms of three stages: sensitization, immediate response, and late phase response. Sensitization is the development of an allergen-specific IgE response in a susceptible individual. The allergen carries out clonal selection of the B lymphocytes. Upon binding through the receptor, B cell internalize the allergen. The dendritic cells uptake allergens and direct naive T cells to become Th2 cells. The net result is allergen-specific memory B cells and IgE. This table shows the effector phases. The sensitization phase results in allergen-specific memory B cells. It also produces mast cells and basophils bound to the allergen-specific IgE through their Fc epsilon R receptors. The second exposure of the allergen results in the allergen cross-linking of IgE molecules on the mast cells. There is an instant degranulation. The biologically active mediator molecules released are shown in the table. The degranulation releases substances which cause vasodilation of blood vessels and attracting eosinophils. Leukotrienes, histamines, prostaglandins and platelet activating factor from mast cells are responsible for immediate reaction of the type 1 hypersensitivity. This slide shows mast cell activation has different effects on the different tissues. For example, immediate reactions in the nose causes redness, itching and increased secretion by mucosal epithelial cells. The late phase reactions are often seen after 2 to 12 hours of immediate reaction. These are caused by mediators of T cell cytokines acting together. A more detailed description of late phase reaction is given in the figure. It is the release of eosinophil chemotactic factor ECFA by mast cells which recruits eosinophils to the site. Histamine and other mediators facilitate their movement by enhancing vascular permeability. Cytokines including granulocyte macrophage colony stimulating factor IL-3, IL-5 cause eosinophil growth and differentiation. After the eosinophilic cationic protein is released, which apart from destroying allergens also causes tissue damage. Similarly, mast cells attracts neutrophils by releasing neutrophil chemotactic factor. Neutrophils degranulate after binding to antibody bound to allergen and release lysosomal enzymes, leukotrienes and PAF. Again, apart from destroying allergens, tissue damage accompanies these activities. To summarize the lecture, we learned that hypersensitivity is of 5 types. Type hypersensitivity is also called allergy. Allergy has 3 stages: sensitization, immediate reaction and late phase reactions. Immediate phase reactions are IgE mediated. Basophils, neutrophils and eosinophils are recruited in the late phase reactions. Degranulation of mast cells, eosinophils and neutrophils releases soluble factors which participates in the overall set of reactions. IgE is vital to the immune defense against parasites, especially worms. The hypersensitivity reactions occur against allergens, which perhaps is an unintended consequence of the IgE mediated responses. The tissue damage is the collateral damage which accompanies the immune response. We will continue discussing type 1 hypersensitivity reactions in the next module as well. Little more before moving over to the description. of type 2 hypersensitivity. Thank you.